Hello friends, welcome to today's session regarding cell, the unit of life. Myself Jalaja Sajeev, PGT Bio from KV Keshapuram. In the previous session, I have already given you to think and solve some of the questions. Now, we can see how many of you have already solved this based on the content, based on the previous knowledge. You can verify your answers with this one. Your first question was, identify which among the following is not correct. A. Robert Brown discovered the cell. Scleeden and Schwann formulated the cell theory. Virchow explained that cells are formed from the pre-existing cells. A unicellular carries out its life activity within its cell. You know which is the correct option? Okay, your answer is Robert Brown discovered the cell. So, this is the incorrect statement among these four options. You know who discovered cell? Yes, it is Robert Hooke in 1665 discovered cell. Other statements all are correct. Now, come to the second question. It was, new cells generate from options A. Bacterial fermentation, B. Regeneration of old cells, C. Pre-existing cells, D. Abiotic materials. You know, new cells will always generate from a pre-existing cell. This was one of the concepts that you have learned in cell theory. A new cell originates from a pre-existing cell. So, your option is C is the correct answer. Good. Now, your third question, that was, which of the following is correct? Options: cells of all living organisms have a nucleus. Both animal and plant cells have a well defined cell wall. In prokaryotes, there are no membrane bound cell organelles. Cells are formed de novo from abiotic materials. So, here your option is, in prokaryotes there are no membrane bound cell organelles. Coming to the fourth question, that is, match the following. You have to match the items present in column A with the items present in column B. Here are the options: A. Cisternae, A. Cristae, B. Cisternae and C. Thylakoids. In B, the options flat membranous sacs in stroma. The second option is enfoldings in mitochondria. Third option, disc shaped sacs in Golgi apparatus. So, try to match this one properly. So, you can see and verify your answers. First one is cristae. Cristae are the infoldings that are produced by the inner mitochondrial membranes. So, infoldings in mitochondria, that is the correct option. Good. Now, coming to the second one, that is cisternae. They are the disc shaped sacs present in the Golgi apparatus. Now, the third one, that is thylakoids. You know, thylakoids are present in the chloroplast. So, they are the flat membranous sacs that are seen in the stroma. Now, your fifth question was, what is a mesosome? And mention the functions. As you have already learned, the plasma membrane in the case of prokaryotes produces infoldings that will fold inwards and forms vesicles, small tubular structures, or even we can say lamellae, and these structures that we will call as mesosome. Functions of mesosomes: since they are infoldings, they will increase the surface area for transport, and they helps in replication of DNA during cell division. Moreover, they helps in the formation of cell wall during the cell division, that is binary fission. It contains some of the enzymes for respiration, so it contributes significantly towards respiration. It helps in secretion. So, these are the different functions of mesosomes. Now, here is the sixth question: how do neutral solutes move across the plasma membrane? Can the polar molecules also move across it in the same way? If not, then how are these molecules transported across the membrane? You can verify your answers. Neutral molecules move along concentration gradient by simple diffusion. That is, the molecules will move from a region of higher concentration to a region of lower concentration till the concentrations become the same. Whereas, polar molecules cannot pass through the plasma membrane by diffusion. It is due to the bipolar nature of lipid bilayer. They are transported across the membrane by facilitated diffusion with the help of some of the specific carrier proteins. They are present on the plasma membrane, and these carrier protein molecules, they will combine with the substrate molecules to be transported, and they are moving across the plasma membrane. Now, here are some of the topics for today's discussion. First comes the structure of cilia and flagella. A cilium or flagellum that has two distinct parts. The first one, that is a protoplasmic sheath. It is the outermost protective coat. It is continuous with the plasma membrane. And the second part, that is axoneme or axial filament. It is the central part and it is formed of 11 microtubules. They are arranged in 9+2 pattern. Here in this diagram you can see the arrangement of these microtubules. In the periphery you can see 9 microtubules and each one is a doublet. And at the center you can see 2 microtubules. So, total in the periphery there are 9 microtubules and at the center there are 2 microtubules. So, there is a total of 11 microtubules present in the axoneme. Now, regarding the types of microtubules, I have already told you at the center they present 2 microtubules. They are the central microtubules, 2 in number, and each one is in turn made up of 13 protofilaments. And these 2 central microtubules, they are surrounded by this central sheath. In turn, when you are observing this diagram, you can see that the 2 central microtubules are interconnected by double bridges. This is the double bridge that interlinks the 2 central microtubules. Now, here you can see the peripheral microtubules. So, the second category of a microtubule is the peripheral microtubule. They are 9 in numbers. You can see here in this diagram 9 peripheral microtubules. And each one is a doublet. You see here, each one is a doublet formed of 2 subtubules. The subtubule is A and B. So, there are 2 subtubules A and B. A subtubule is slightly inner in position and is formed of 13 protofilaments, whereas B subtubule is slightly outer in position and it is formed of 10 globular protofilaments. So, A subtubule made up of 13 protofilaments, B subtubule made up of 10 protofilaments. So, in this way each microtubule is a doublet. A tubule is linked with the B tubule. In each of the doublet, A tubule is linked with the B tubule with the help of linkers, bridges, that they are known as AB linkers or interdoublet linker. A subtubule is connected with central sheath with the help of radial spokes. Here you can see in each doublet the A subtubule that is connected with this central sheath and it is with the help of radial spokes. This is the radial spoke. Here you can see the enlarged view of the same diagram. 9+2 arrangement of microtubules 9 microtubules in the periphery, and each one is a doublet with A and B tubules, subtubules. A contains 13 protofilaments, B contains 10 protofilaments. A is linked with this central sheath. You can see this part as central sheath. Then here you can see at the center 2 tubules interlinked. Now coming to the differences between cilia and flagella. The internal structure remains the same. Now regarding its differences, cilia, that is more in number compared to flagella. That means flagella is less in number. Then regarding its size, cilia is smaller in size and flagellum is larger in size. Generally, cilia are seen all over the body surface, but flagellum is seen located at the anterior end of the organism generally. And regarding the movement, they helps in sweeping or even rowing movement. Here flagellum helps in undulatory movement. So there is a slight difference in this movement. Then coming to the different other functions, it helps in locomotion. Cilia help in locomotion and feeding and circulation. Flagellum, that helps in locomotion only. So here you can see the differences even though the structure is same. Now coming to mitochondrion, at another important cell organelle for a eukaryotic cell. It was first discovered by Kolliker in flight muscles of insects, and the term mitochondrion was coined by Benda. It is present in all the eukaryotic cells except in mature mammalian RBCs. You know, it is absent in all the prokaryotic cells. Now regarding its shape varies from granular, spherical, oblong or even discoidal. Now regarding the size of the mitochondrion can also varies from cell to cell depending upon the metabolic status of the cell or the function of the cell. If a cell is metabolically very active, then it contains large number of mitochondrion. Number will also varies with respect to the metabolic status of the cell. They are seen more in numbers in a metabolically active cell as well as in a growing cell and in a dividing cell. Maximum number of mitochondrion are found in a flight muscles because they need more amount of energy. So, more respiration, more oxidation. So, it needs more number of mitochondrion. Now regarding its structure, you can see the diagram here. It is a double membrane bound organelle. There are two membranes, one is the outer membrane, it is already labeled here, outer membrane, then there is the inner membrane. So, in this way there are two membranes, it is a double membrane bound organelle, and when you are observing, you can see that between these two membranes there is a space, a narrow space is there that we will call it as perimitochondrial space. Now, there is one more special thing, interesting one, when you observe this inner mitochondrial membrane, here you can see finger like or tubular projections, and these tubular or even finger like projections, that we will call it as cristae. It is needed for increasing the surface area of the metabolic activity, respiration rate. Now, again you can see some stalked particles here attached with this cristae. So, they are known as F1 particles or fundamental particles or elementary particles, or we can call them as oxysomes. Then the inside this mitochondrion you can see fluid like structure, and that one is known as the matrix. It is called as matrix. When we are observing this matrix, we can see a circular DNA, cDNA, small c and DNA, that means circular DNA. It is double stranded, contains genes. Now, in addition, it contains ribosomes. Here the ribosomes are 70S ribosomes. We will call these 70S ribosomes as organellar ribosome, or we can say they are the prokaryotic ribosomes. Then in addition it contains ATP molecules, mineral ions, protein molecules, etc. So, these are the various points regarding the structure. So, you can see here once again it is a double membrane bound organelle, or each membrane its chemical composition. It is made up of lipoprotein in nature, just like that of plasma membrane, made up of lipids, phospholipids and protein molecules. And in between these two membranes, as I have already told you, there is a narrow space, and that narrow space we will call it as perimitochondrial space. The inner membrane is producing a finger like enfoldings, and these finger like enfoldings we will call it as cristae. You know, the function is increases the surface area for cellular respiration. Now, regarding the number of cristae, length of the cristae, size of the cristae, that depends upon the size of the mitochondrion as well as the metabolic rate of the cell. The M phase, what is M phase? Matrix phase of the cristae that contains many knobed particles, stalked particles, and they are known as the F1 particles. We can call these F1 particles as fundamental particles or even elementary particles. They are also known as oxysomes or electron transfer particles. They helps in electron transport. Now, here you can see the structure of an F1 particle. Here you can see, as I have already told you, they are present in the M phase of this cristae. M phase means matrix phase. And here you can see it is a stalked particle with 3 subunits. The basal piece or base piece, also known as F0 particle. Then there is a small stalk. It is known as F5, F6 subunit. At the tip portion, terminal portion, you can see a globular head piece, and this head piece is known as F1 subunit. Head piece helps in the synthesis of ATP. For this purpose, there needs the enzyme, and for the synthesis of ATP, there needs the enzyme ATP synthetase. In short, we will call it as ATPase. So, ATPase enzyme is seen in the head region, and the function of these F1 particle is they helps in the synthesis of ATP molecules during cellular respiration. So, here already I have told you it possesses 3 parts: base piece or F0 subunit. It is rectangular in shape and it is kept embedded inside the or inner membrane. Then there is the second part that is called a stalk, known as F5, F6 subunit. It interconnects the head and the base piece. The third portion, that is the head piece, we will call it as F1 subunit. It is spherical in shape and it contains the ATPase enzyme or even ATP synthetase enzyme needed for the synthesis of ATP molecules. Inside the mitochondrion, there present a dense granular substance, fluid matrix, and it contains various enzymes for Krebs cycle. Then it contains enzymes for the synthesis of different types of proteins and lipid synthesis. It also contains 70S ribosome, the prokaryotic ribosome or the organelle ribosome. We will call it as mitoribosome since they are present inside the mitochondrion. In addition, it contains circular DNA. We will represent it as small c DNA, and there present mineral ions. Mitochondria is a semi-autonomous organelle or a self-replicating organelle, as it contains 70S ribosome, the circular DNA molecule and ATP molecules, which are highly essential for making some of the proteins. Since they are making their own protein meant for replication of DNA, it is known as a semi-autonomous organelle or a self-replicating organelle. Now, coming to the functions of mitochondrion. Mitochondrion are known as powerhouse of the cell. It produces, it stores, it distributes energy in the form of ATP molecules. Moreover, it helps in protein synthesis because all the machinery needed for protein synthesis is present inside the mitochondrion. So, it helps in protein synthesis. Since it contains DNA, it can control characters also. There is a phenomenon that is cytoplasmic inheritance. So, this particular DNA that contributes to the cytoplasmic inheritance of characters. Now, come to the next organelle, that is plastids. The term plastids, it was given by Haeckel. They are found in all the plant cells and in some of the protist organisms, for example, euglenoids. Types of plastids based on pigment molecules. There are 3 different types of plastids based on the pigment molecules. The first one is leucoplast. You know, leuco means colorless or white. So, they are known as colorless plastids. They do not have any granule. So, agranule. They do not have any chlorophyll, they are colorless. So, it is known as agranule, and they are not exposed to sunlight. That means they are seen in parts that are not exposed to sunlight. For example, roots, they are seen in the rhizome, they are seen in the seeds. It is seen in all the colorless parts of the plants which are not exposed to sunlight. They help in the storage of food molecules. Here comes one of the application level question. If this type of plastids present in the roots or in the rhizome when exposed to sunlight for a longer period of time, what will happen? They will change their color, they absorb light and they will change their color and they will transform into chloroplast. So, in short, colorless plastids without any chlorophyll and granum, seen in the parts that are not exposed to sunlight normally, such as roots, rhizomes, seeds, and even in some of the flowers also you can see. So, if they are exposed to sunlight, they can be transformed into chloroplast. Now, here comes the chromoplast, colored plastids. Chromo means colored, colored plastids other than green. They are seen in the leaves, even in flowers, fruits, etc. They help in pollination. They help in dispersal of fruits and seeds to distant places, and they even helps in photosynthesis. They can act as accessory pigments. They contain pigments which can act as accessory photosynthetic pigments. They will help in absorption of photons and contributes to photosynthesis. The third one, that is known as chloroplast, green plastids. No, they contains chlorophyll, well developed granum, absorb photons, so helping in photosynthesis. Now, there is yet another classification of plastids based on their storage product. Which storage product they are storing? So, based on that we can again classify these plastids into 3 different types. The first group amyloplast, second one alleroplast, third one aleoplast. First category, amyloplast. Amylose refers to starch. So, these are plastids that store starch. So, they are found in the storage parts, for example, potato tuber. wheat, rice grain. Second one is alleroplast. Alleron grains means protein grains. So, these are the plastids that stores proteins. They are seen in maize grains, pulses, etc., where abundant amount of proteins are stored. The third one, that is oleoplast, they will store fat, and they are found in some of the seeds like castor, mustard and groundnut. Now, coming to the structure of chloroplast, yet another double membrane bound organelle present in all the plant cells, eukaryotic cells. They are discovered by Anton von Leeuwenhoek, and the term chloroplast was given by Schimper. They occur in all the photosynthetic plants and even in euglena also. Their shape varies from cup shaped, they may be spiral, discoid, oval or reticulate. In short, their shape can vary. Similarly, the size will also vary from cell to cell. They are again double membrane bound organelle. You can see here the diagram structure. They are also double membrane bound organelles with the two membranes, outer membrane and this one inner membrane. So, two membranes you can see clearly here. Now, regarding its chemical composition, regarding its chemistry, it is lipoprotein in composition, just like that of our plasma membrane, made up of phospholipid and protein, and it is trilaminar in nature. As in the case of mitochondrion, here in chloroplast also you can see a small narrow space between the two membranes, that is called as periplastidial space. So, this is the periplastidial space, the narrow space seen in between the two membranes. Inside the chloroplast, there present a dense granular substance. Here we will call this dense granular fluid substance, the fluid matrix, that as stroma. So, this dotted portion, the fluid matrix, we will call it as stroma. This one we have represented as stroma. It also contains 70S ribosomes. These red colored dots, spherical ones, ribosomes. The type of ribosome here is 70S ribosome, just like that of mitochondrial ribosome. Here also you can see circular DNA, cDNA, circular DNA. Then they present a messenger RNA, small mRNA means messenger RNA, different types of enzymes and tRNA. tRNA means transfer RNA molecules and mineral ions. So, all these are the components that we can see inside the stroma. When we are observing this inner membrane, inner plastidial membrane, it produces so many invaginations. They are invaginated to form layers, lamellae, and since these lamellae they are seen in the stroma, we will call it as stroma lamellae. You can see here the labeling, stroma lamellae. It is an invagination of the inner plastidial membrane. Now, the interesting thing here is when you are observing this stroma lamellae, here and there they will enlarge to form a disc like structure. They are enlarging to form a disc like or even a sac like structure which is known as the thylakoid, the individual one that we can say as thylakoid. And see, these thylakoids are arranged one above the other, just like that of a pile of coins, and this particular single hole structure obtained by placing the thylakoids one above the other, that structure we will call it as granum. You can see here in the diagram granum. So, what is the unit of granum? Yes, the unit of granum is thylakoid. And what this thylakoid contains and how many thylakoids are there normally? It can also varies. A granum contains 10 to 100 thylakoids. And inside the thylakoid, so the unit of a granum is thylakoid, and inside that thylakoid you can see small round paracrystalline bodies. And these small round paracrystalline bodies, they are known as quantosomes on its inner membrane. And these quantosomes, you see, it contains 200 to 300 chlorophyll molecules. So, the granum contains chlorophyll molecules, or rather the thylakoid that contains the chlorophyll molecules, which will trap the photons, light particles for photosynthesis, for light reaction. So, here comes stroma helps in dark reaction and granum helps in light reaction. Why it is so? Granum made up of thylakoids contains quantosomes which contains chlorophyll molecules that can trap the photons, helping in light reaction, whereas stroma contains different types of enzymes needed for dark reaction. So, quantosomes can absorb photons for photosynthesis. Now, chloroplasts are also semi-autonomous organelle or self-replicating one, just like that of mitochondrion, as it contains the complete machinery for the synthesis of some of the proteins. But for the synthesis of some other proteins, they are to depend upon the nuclear DNA and the cytoplasmic ribosomes. Now, coming to its function, the first and the primemost function is photosynthesis. It will trap light energy and it helps in photosynthesis. As a result of photosynthesis, there is the evolution of oxygen. You have already learned the same in photolysis of water in the chapter photosynthesis. Then it is needed for protein synthesis. It helps in protein synthesis. Since it contains circular DNA, it helps in hereditary function also. Here also it can contribute to the cytoplasmic inheritance. Now coming to ribosomes, isolated first by Claude from the cell cytoplasm. Robinson and Brown reported it in a plant cell. The term ribosome was coined by G. Pallade. They are ribonucleoprotein particles seen freely in the cytoplasm, or they may be attached with some of the organelles, they may be attached with the nuclear membrane or rough endoplasmic reticulum, or they may be seen or they are seen inside the organelles like mitochondrion and chloroplast. As we all know that they are known as universal organelle, since they are present in all the plant and animal cells except in mature sperm as well as mature mammalian RBCs. Each ribosome has two subunits. Ribosomes lack any outer membrane, so we can call them as subcellular organelle or even a naked organelle. Number of ribosomes depends upon the RNA content of a cell. They are seen more in the case of liver cells, meristematic cells, cancer cells, and even in endocrine cells. Now regarding the types of ribosomes, there are two different types of ribosomes. One is 70S ribosome and the other is 80S ribosome. Now regarding the 70S ribosome, 70S ribosomes found in prokaryotes and in the organelles of mitochondrion and plastids. Each ribosome has two subunits. Here the subunits of a 70S ribosome is 50S and 30S. What is this S? S stands for Seybertberg unit or sedimentation coefficient. Now, when we observe this 50S subunit, the larger subunit, it contains different types of RNA and different types of proteins. In the 50S subunit, that means the larger subunit of 70S ribosome, you can see 23S RNA, 5S RNA and 34 different types of proteins. Now, coming to the smaller subunit, the 30S subunit of a ribosome, 70S ribosome, it contains 16S RNA and 21 different types of proteins. Now regarding the 80S ribosome, I have already told you there are two types of ribosomes, one is 70S and 80S. Now regarding the 80S ribosome, it also has two subunits such as 60S and 40S. 60S is the larger subunit and 40S is the smaller subunit. 60S subunit has three different types of RNA you can see here, 28S, 5.8S and 5S type of RNA and 40 different types of proteins. Now coming to this smaller subunit, that is the 40S subunit. It has 18S RNA and 30 different types of proteins. Normally, in a cell, these subunits are seen separated, and at the time of protein synthesis, these two subunits, they will associate together to form a single ribosome, and the association of these ribosomal subunits depends upon the magnesium ion concentration. A specific concentration of magnesium ion is needed for the formation of a whole ribosome. Now here comes the term polysome or polyribosome. What is meant by a polysome? Poly means many, some here means ribosome. So it is an association of many ribosomes on a messenger RNA, and it is formed at the time of protein synthesis. Now, the distance between two adjacent ribosomes in the case of a polyribosome will be 90 nucleotides apart. Coming to the functions of ribosome, they are helping in protein synthesis. So it is the organelle for protein synthesis. Free ribosomes produce non-secretory proteins, for example enzymes for intracellular use. Bound ribosomes present on the rough endoplasmic reticulum synthesize secretory proteins, that means enzymes for extracellular use. Now here are some of the questions for you to think and answer. Name two cell organelles that are double membrane bound. What are the characteristics of these two organelles? State their functions and draw the labeled diagram of both. Next question, what are the characteristics of a prokaryotic cell? The third question, multicellular organisms have division of labor. Explain. Fourth one, cell is the basic unit of life. Discuss in brief. The last question, both lysosomes and vacuoles are endomembrane structures, yet they differ in their terms of their functions. Comment about it. Thank you.